If you're a student in microbiology, trying to identify bacteria from the Enterobacteriaceae family can feel, well, a little overwhelming. It's a huge group, and so many of them look the same under the microscope. But don't worry. The goal of this explainer is to break it all down into a simple, logical process, one step at a time. Okay, so it all starts right here with a slide. You've taken a sample, you've stained it, and under the microscope, this is what you see, a whole field of these little pink or red rods. This tells us they're gram-negative bacteria, and that's our first big clue in this diagnostic mystery. But right away, we have our first big problem. Is this E. coli? Is it Salmonella? Maybe Proteus? Just by looking at them like this, there's no way to tell. They all look like medium-sized gram-negative rods. So, how in the world do we figure out which specific culprit we're dealing with? Here's the game plan. We'll start by meeting the usual suspects. Then, we'll ask one big question that helps us narrow them down. After that, we'll learn how to read the plates for more clues, and then finally confirm the identity. And at the end, we'll talk about why all of this really matters. All right, first up, let's get properly introduced to the family that we're investigating. The Enterobacteriaceae are a huge and really important family of bacteria. If you're in a veterinary lab, these are the guys you are going to see all the time. Now, while many of them are just normal residents of the gut, some are pretty serious pathogens, and that's exactly why identifying them correctly is so critical. So all members of this big family share a few core traits. You can think of this as their family signature. They are all gram-negative rods, they're catalase positive, and, this is important, they are oxidase negative. That oxidase test is a key step. If you have an organism that's oxidase positive, it's not in this family. So these tests just confirm we're on the right track. Okay, so once we're sure we have a member of the Enterobacteriaceae family, we get to ask the single most important question to start narrowing down our suspects. Can it, or can it not, ferment lactose? Now, to answer that question, we use a special tool called differential media. This might sound a little complex, but it's actually a really clever idea. It's basically a special kind of auger that's designed to take invisible differences, like whether or not a bacteria can eat lactose, and make them visible to our naked eye, usually with a color change. And the classic differential medium for this family is called McConkey agar. Let's just quickly break down how it works. First, it has inhibitors in it that stop gram-positive bacteria from growing. This basically clears the field for our suspects. Second, it's got lactose as the main food source. And third, it has a pH indicator called neutral red that's going to act as our little color-changing informant. So this slide shows the two possible things that can happen. Look on the left. If the bacteria can ferment lactose, they produce acid. This makes the pH drop, and that indicator dye turns a bright hot pink. Now on the right, if they can't use lactose, they eat peptides instead. This produces basic byproducts, and the indicator just stays pale. This difference right here is a really crucial point and something you'll definitely want to remember for your exams. And this just illustrates that difference perfectly. Look here. On the left, you see those unmistakable bright pink colonies of E. coli. It's a strong lactose fermenter. And on the right, you see the pale, almost colorless colonies of Salmonella, which is a non-lactose fermenter. Just this one test has split our huge list of suspects right in half. So here's a simple rule of thumb that's incredibly helpful to remember. When you see pink on that McConkey plate, your mind should immediately jump to the big lactose fermenters like E. coli and Klebsiella. If those colonies are pale, you should start thinking about important non-fermenters like Salmonella and Proteus. So color gives us a huge clue. But beyond just the color, we can actually get more information just by looking at how the bacteria are growing on the plate. Some of these species have very distinct personalities. Take a look at this blood agar plate. Do you see those wave-like rings that are spreading out from the center? It almost looks like ripples in a pond. This is called swarming, and it is a classic sign of Proteus. This bacteria is supermodal, and it literally just spreads itself across the surface of the agar in waves. If you see this, it is a massive clue. Another great visual clue comes from Klebsiella pneumoniae. Now, it's a lactose fermenter, so its colonies are pink, but they also look different. They're often very large, and they look really wet and slimy. This is because Klebsiella has a super thick capsule made of polysaccharides, and that's what gives the colonies this mucoid or slimy texture. And then you have some bacteria that just like to show off. Certain strains of Serratia marcescens produce this beautiful, distinct red pigment. And that color isn't from a pH indicator. That's the bacteria itself making it. You tend to see this most often when the plate is incubated at room temperature.
Visual clues are fantastic, but to be absolutely sure about our suspect's identity, we need to run a few final biochemical tests. These tests basically check for specific enzymes and metabolic pathways. Let's use E. coli as our main example here.
A really classic set of tests is called the IMVC series. The name is just an acronym for the 4 tests: indole, methyl red, Vos for scour, and citrate. A typical E. coli strain has a very predictable pattern. It's positive, positive, negative, negative. You can think of this as its biochemical fingerprint, and it's a great way to get a quick presumptive ID.
Now let's connect that chart to what it actually looks like in the lab. For E. coli, we said the I for indole is positive. Well, on this slide, you can see exactly what that positive test looks like. After you add the reagent, notice that bright red ring that forms right at the top of the tube. That ring is our visual confirmation that the bacteria has the enzyme it needs to break down tryptophan into indole. So, we've gone through all these steps to put a specific name to a bacterium. But why? Why is this so important? Well, this is the part where we connect all the dots from the lab bench directly to the health of the animal.
Because honestly, not all bacteria are created equal. Just look at this table. Being able to distinguish E. coli from Salmonella is absolutely critical. While a lot of E. coli strains are just harmless gut residents, some strains are very serious pathogens. Salmonella, on the other hand, is almost always considered a pathogen. Knowing which one you're fighting dictates everything: the treatment, the biosecurity, and even the prognosis for the animal. And this diagram really shows us the stakes. A pathogenic E. coli, like the one shown here, can go from being normal gut flora to colonizing mucosal surfaces, and then passing right through the gut lining and into the bloodstream. From there, it can cause systemic disease, leading to serious infections like mastitis or even shock and death. Each of these organisms has its own unique way of causing trouble. So we started with just a simple pink smear on a slide, and we followed a logical path of clues to get to a specific identification. This whole process is a really powerful diagnostic tool. So the final question is for you. Now that you can tell these bacteria apart, how are you going to use this step-by-step knowledge to help diagnose your next